In 2018, our research group started work on building knowledge of the exotic fly spotted-winged Drosophila, which is also known as Drosophila suzukii. This pest is becoming a pest of concern for berry and stone fruit industries around the world. Throughout this project, we've been working towards a goal of increasing preparedness of our plant industries for a possible incursion of spotted wing, as well as increasing our capability to respond to a detection should one occur. The research team has been collecting information and knowledge on surveillance and control methods and has also been developing models which will help us predict how well Australian regions and conditions will support spotted-winged Drosophila, and this will give us an idea on how far this pest could spread and establish. Educating potentially affected industries has also been a really crucial part of the project because it will be critical if we're going to achieve early detection of spotted wing and also allow us to respond to this pest should it arrive. This video tutorial has been developed to educate you, your colleagues or your students on the importance about spotted wing. This research has been a collaborative effort between Plant Health Australia, Plant and Food Research New Zealand and CSER, and has been made possible with funding from Hort Innovation, using the strawberry, raspberry, cherry, blackberry and summer fruit research and development levies, and also contributions from the Australian Government.
Hi there. Today I'm going to speak about Drosophila suzuki, the spotted-winged Drosophila, particularly the incursion process, both at a global and local scale, the pathways of movement, some of the trapping options and potential economic impacts. Now, the work we've done has been with support of B3, Better Border Biosecurity, Horticulture New Zealand, and Hort Innovation. Now, just looking at its global movement, so it's native to India and through to Japan, so quite a large area. In 1980, it managed to make its way to Hawaii, where it was quite content for many years. Then in 2008, it was found both on mainland USA and in Europe. And studies have found that it has quite a high genetic diversity in both the European and mainland US populations, which suggests that there have been multiple invasions or incursions of Suzuki into these areas. Now in 2011, it was found in Mexico, and 2013 Brazil, and from there it has moved into other countries. Now, I'd just like to acknowledge Ruth and Carlos here from SAG in Chile, and they've shared some of their data on what they experienced when Drosophila suzuki turned up, or how quickly it moved through the country. So they have been monitoring since 2012 throughout a few different areas in Chile. And it wasn't until 2017 that they had their first detection, which sparked a lot more incursion investigation, and it was discovered in a reasonably wide area. Now in 2018, it had moved quite a way north in the country, along those growing areas there, which sort of runs along the centre there. And the two maps on the screen represent about 600 kilometres each in height. So you can see that it's moved, it's found over 900 kilometres apart in the space of a year. Now what that looks like in terms of catch, on the graph here we can see in May 2017 there was a very small catch of Suzuki, and then in those same areas the following year, massive peak or increase in catch. So how that translated to catch of insects and traps. In 2017, the ratio of spotted-winged Drosophila to other Drosophilas that were attracted to the trap and lure was 1 spotted wing to 600 other Drosophila. Fast forward a year, same place, same time, the ratio is now 1 to 1. You can see at the bottom left-hand corner there, there's a picture of the trap that's used for this trapping. So this is the BioBest trap with the BioBest lure. And inside that trap you can see there's a liquid. So the flies are attracted to that and then they get stuck and drown. once in the country, how do they move about? Or even globally, how would they move about? So they're similar to a fretted fruit fly in the way that their larvae are inside fruits. It's the most likely way it's going to move around the world and in the country. And we found from looking at some of the European information and data that quite often Drosophila suzuki was found in picnic sites or truck stops and those sort of places. You can imagine someone's driving along at the stop, they've eaten some fruit and thrown away the leftovers, or perhaps they've noticed that their fruit looks a bit off and thrown it out. Now it has a very wide host range of both commercial and non-commercial fruits. And here's a picture of some strawberries that have been deliberately infested for a trial. Now from earlier work, we know that if we can detect an insect early on, we have an improved likelihood of eradication success, or at least better management options should we not be able to eradicate. Now this data comes from the Global Eradication Response Database, which is freely accessible, and there's the web address there. And you go and register, access a lot of the data and contribute your own. And we found that if you have a lure, at least for the eradications, there's a 20-fold increase in the probability of having a successful eradication attempt. So how are we trying to detect these in New Zealand? Now of course we have a number of import health standards that we must follow, and then there is information on what can be imported when, where, and what sort of treatments must be assigned to that commodity prior to being let into the country. Now on top of that, there are also checks at the border, and there was a detection of spotted-winged Drosophila in an orange in a consignment from the US in 2019. Prior to that, there was a detection in 2012 out of Asia. Now, there's also general surveillance run in New Zealand. So this is in terms of public or industry calling a free phone number and talking about what they've seen, what they've found. They may have to send a photo and the like, and that may be investigated further if it's a biosecurity risk. And that phone number gets around about 10,000 calls per year. But currently there's no active surveillance, so no traps that are placed out prior to detection of spotted-winged Drosophila. But there are a number of lures and traps out there. Now we'll start with some of the better ways to sample for larvae. So of course you can open your fruit and have a look. But there is a flotation technique which has been developed. And here's a good video to have a look at this, and it's developed by colleagues at EMR in the UK. So instead of opening fruit and maybe squishing your sample or somehow missing the larvae, what you can do is put a number of fruit in a bag, break them open slightly, and then add a high concentration of sugar water to that and let the fruit sit in there. And this encourages the Suzuki larvae to exit the fruit, and then they rise to the top. You can see them there along the top of the water. But I suggest you watch the video to get some more information and also the recipe for how to do this. So there are a number of trapping options available for Drosophila suzuki. These can be both selective and non-selective, so ranging from catching primarily Suzuki plus some bycatch to attracting a large range of insects, as well as synthetic and non-synthetic lures. So examples of some non-synthetic lures are ones that are vinegar based or for fermenting products. So these tend to attract a large number of species to the trap beyond just Drosophila suzuki, and will have a shorter life of the lure than, say, a synthetic lure. And these have also been developed over the past few years and range in their selectiveness and attractiveness. These tend to last a little bit longer as well in the trap and can be used with a drowning solution or potentially a dry trap with the right sort of killing agent in the trap. And there's a range just shown on the screen. So here's what you can expect to get in some apple cider vinegar. So here's a trap we had out. You see some light brown apple moths in there, a lot of ants, looks like some cockroaches. So pretty low selectivity. Very cheap though, and quite easy to get. You just go down to the local supermarket and buy it and put it in a trap. Now the trap needs, the liquid needs weekly replacement. So that one is because you get so many insects and they start going off, but it also can evaporate quite quickly in warm weather. Now the BioBest lure and trap, that is also a non-synthetic lure, so it's probably not going to have any regulatory issues. And there are suppliers of this in both New Zealand and Australia. The lure replacement is recommended every fortnight, and the lure should be combined with this trap system, so this red round trap, and you can see these three circled areas where the flies get into the trap. They are to reduce the size of the insects that they can get in. So a lot of other insects are still attracted to this, but because of the little filtering system, smaller insects get into the trap. In terms of synthetic lures, it could be a wet or dry trap, so it's recommended that you have a drowning solution, but with the right sort of killing device, there shouldn't be any reason why you couldn't have a dry trap, which may make it a bit more pleasant for the diagnosticians to be going through the traps, similar to what you could expect in, say, a fruit fly trap. Now the lure replacement is recommended for every 4 to 6 weeks. It lasts a lot longer. It is a bit pricier, of course, and there also is the potential for some regulatory issues because we're dealing with compounds that may have requirements under local law of how much can be used, when, how often, and with what. Now here are some lures that are available. So you've got Alpha Sense and Century and Trace A. So the Trace A HS is a high specificity, so reduced bycatch and reduced catch overall. And then you've got the Trace A BS, which is a broad spectrum, so much larger lure, a lot more compounds being released, and a lot more insects being trapped. So once you get something in a trap, how are you going to tell whether it's Suzuki or not? So what the males look like, spotted winged Drosophila, so they live up to that name. So you can see the males have, quite often, have 2 dark spots on the ends of their wings. And of course, they don't always have it, but most often do. It can be various shades as well, quite dark to quite light. And that can be quite easy to see with the eye. And if you've got really good eyes, you might be able to tell the females from the males, or the female Suzuki from others. But you'll probably need at least a microscope. So Drosophila melanogaster and other Drosophilas tend to lay their eggs in decaying fruit or ones that have already been damaged. So when damaged, and there's an opening and they can deposit their eggs in there. Whereas Drosophila Suzuki has this sclerotized, or the stronger part of the body that can cut through the ripening fruit. So you can see the little teeth there along the edge, and they can put their eggs directly into that ripening fruit while it's still on the tree. Now you may have really big samples or a larger number of flies in there, and people have been developing molecular tools to try and distinguish Drosophila Suzuki from everything else that is in there. And there has been quite a bit of success, and there's a paper there you can read and have a look at. And what people are trying to do now is trying to develop methods that can sample a large number of traps at a time, or give you a result reasonably quickly, rather than having to go through quite a long process. So it becomes quite a helpful tool for the people in the field checking these traps. So how many traps do we have to place out there to catch spotted winged Drosophila? Now we've estimated the effective sampling area or the ESA, of a Sentry lure and trap, and this trap is a red panel trap. And it comes from work published by Danielle Kirkpatrick in 2018. And in that trial, in the work, she did some mark-release recapture trials, which gave us an estimate of how effective the lure was at recapturing flies at different distances from the release point. Now, as you'd expect, as you move further and further away from the source of the lure and the trap, that your probability of catching a fly or a fly being attracted to that trap is reduced, and you get this curve. Now, our curve is quite hard to model, so what we can do is push that into a cylinder shape, which is a uniform shape of 1 unit in height, and that top of the cylinder shape is the effective sampling area. So a large one, a large top, would have a high effective sampling area, so over quite a wide range, and a small one. Now, that's not the probability of being trapped, but it's a correlation coefficient that can be used later on to estimate a probability of detection. Now, using that Sentry lure, or the information published from there, we have estimated the probability of detecting spotted wing Drosophila populations of different density with different numbers of traps per hectare, so population density and trapping density. And as you'd expect, the more traps you place out in the area, the higher probability per day, this is a daily probability, of you detecting that fly, so catching at least one of them in a population. Of course, the bigger that Suzuki population is, the more likely are you to trap it. Now, when you get down to 1 trap per hectare, you have a very low likelihood of trapping or detecting one of those flies. It could still happen, but it's pretty unlikely. So that's looking at the probability on a daily basis. So if we start trapping over multiple days, that probability of detecting that population increases. Now, here we have a hypothetical population of 10 flies that are trappable, and the trapping happens over consecutive days. So as you'd expect, the higher trap density leads to a high probability of detecting a fly, and then after that a response can be mounted. And the 100 traps and 50 traps are actually pretty close. We start seeing a bit of a lag in detection once we hit 10 traps and 1 trap per hectare. Very low. It's not until you've gone through almost half the year that you have a more than 50-50 chance of detecting that fly, or a fly from that population. So what are those potential crop hosts out there? Now, Dave Bellamy and colleagues in 2013 looked to develop a host potential index based on 4 key parameters, those being the attraction of flies to the odours, to the smells of the fruits, the ability of individuals and the ability of populations to oviposit in those fruits, and then the development of the larvae in the fruit. And what they found was raspberries and strawberries are quite attractive. But you can see there's quite a big range in the host potential of strawberries, whereas we can see down at the other end, so least preferred out of the ones tested were grapes. Now, looking at what's happened overseas, we've observed that much of the economic impacts have been driven primarily by the insects directly attacking the fruit, laying their eggs in there, and the larvae develop and make that fruit unsellable. Now, on top of this, there are secondary effects that can impact on those fruit. So as the females oviposit, some of those larvae may not survive. However, that hole is then in the fruit for fungi or rots and the like to then get into the fruit and again reduce yield. Now, in terms of losses that have been observed overseas, in 2016 in the US, $700 million worth of crop loss was attributed to spotted wing Drosophila. And the strawberry industry in Brazil saw 30% loss in their crop. And looking to Italy, it's in the northern area of the Trentino region, they had a range of 25 to 35% berry reduction, and this depended on the crop, was due to Drosophila suzuki. Now on top of that, some of the management practices have required some additional sprays, and this has then interrupted integrated pest management strategies that are already present in the system. So this is sprays killing the biological control agents that have been attacking other pests of the crop. And because these biological control agents have then been disrupted, there is a resurgence of those previously managed pests. However, it's not all doom and gloom. It's been seen and observed that changing some practices such as orchard hygiene, so removing all the fallen fruit or cleaning up after the end of the season, can reduce the density of Drosophila suzuki in the crop. Now on top of that, some changes to harvest practices have also reduced that impact. So going to a more regular regime or faster picking regime with fewer days between harvests has reduced the number of fruit that are then available for Suzuki to attack. So what is at risk should spotted-winged Drosophila arrive in a country? And here we have some information from New Zealand from the 2016-17 year on the value of various host crops for both the domestic and export markets. So we're looking at around about 113 for the domestic market and 120 million for the international market could be at risk. However, we should take heart of what's happening overseas in terms of managing spotted wing. So we've seen that changes to hygiene practices or harvesting times can reduce the impact that spotted wing Drosophila can have on a grower. So thank you for your time. If you want any more information, feel free to contact me at that address. And long may we keep spotted wing Drosophila out of New Zealand and Australia. Thank you.